Welcome, everyone. My name is Jeff Stallman. I'm the Fungus Conservation Coordinator at the Global Center for Species Survival, and good morning from Indianapolis, Indiana, in the USA. You are in and have been hearing the planning of the uh, webinar on aquatic fungal diversity for the International Day for Biodiversity in 2026. We are happy you are here, and I'm going to share my screen. So, the Global Center for Species Survival is one of the co-organizers of this event. We support activities within the International Union for the Conservation of Nature's Species Survival Commission in partnership with the Indianapolis Zoo. We are a team of 7 conservation coordinators aligned with conservation committees within the IUCN SSC, and we support those conservation committees and specialist groups, including the Aquatic Fungal Specialist Group within the IUCN SSC. If you're interested in conservation activities within the SSC, I'll put my contact information in the chat, and I'd be happy to talk with you about fungal conservation or connect you with any of the other conservation coordinators. And now I'd like to pass things over to Dr. Jennifer Anderson to introduce one of the other co-organizing organizations.
Hi, everyone. My name is Jenny, and can you flip the slide for me? Thanks. And it's my pleasure to talk to you about the work we're doing in Fun Action, where we're working towards the conservation of aquatic fungi. So in Fun Action, we have 3 kind of main goals, and that is we summarize as foundation, conservation, and communication. So we are working on producing foundational knowledge needed to form the basis of aquatic fungi-aware conservation. We are committed to translating that knowledge into conservation planning, and we're working to communicate about these fungi and their conservation and their ecological importance to as broad of an audience as possible. I invite you to learn more about us on our website. Jeff, if you want to flip the slide. Thanks. Or you can contact one of these partners.
Thank you. Maybe I introduce the other organizing, project, let's say, in for today's event. So we are hosting that also from the perspective of Most Fun, which is another Biodiversity Plus project. And the focus and the goal of Most Fun is to add aquatic fungi to routine biodiversity monitoring programs. And the way we plan to do that is to collaborate a lot with projects and programs which we exchange as samples and data and experiences, but that these projects and programs focus on other organism groups than aquatic fungi. But nevertheless, their data, samples, and experiences are, of course, valuable and useful to develop tools and strategies that then also cover aquatic fungi. And so these topics that we cover today, they're of course all also relevant for that perspective for biodiversity monitoring of aquatic fungi and their roles in the ecosystems. So I'm looking forward to this. Maybe you can also flip the slide, Jeff. There should be a second one. Yeah, so that's basically an icon that summarizes everything I just said. So it goes from data collection, which we actually also do as part of the project. So not only exchanging that with external projects and programs, but also adding our own sampling campaigns in understudied ecosystems. With this, I think I said everything I wanted to do. You have more information if you scan that QR code up there about the project, and I would like to hand back to Isabel, actually, to introduce another co-organizer of the event today. Thank you.
Thank you, Andy. Hello, everyone. It is my honor and pleasure to represent today the IUCN Species Survival Commission Aquatic Fungi Specialist Group. The group was formed at the end of 2023, and our vision is to develop the scientific and coordination basis for the first conservation assessment, planning, and action efforts for aquatic fungi. The group has now 27 members from 20 countries, and we are open for any person willing to work towards aquatic fungi conservation. So you have more information on the QR code there. And that's it for the short introduction from the group. I will now move to say just a few details about how the webinar will go today. So we will have 4 speakers lined up for today. And the speakers will have about 10 minutes to make a short presentation about different topics. And then there will be about 5 minutes time for each of them to have a question and answers to answer your questions. So please put any questions that you have for our speakers today on the Q&A setup from Zoom. And without no further ado, I will start by introducing our first speaker of today, which is Dr. Prabha Pant. Dr. Prabha Pant is working as a guest faculty in the Department of Botany at the Kumon University, Nainital since 2020. She also worked as a junior research fellow at the Department of Biotechnology sponsored project. Her research interests are aligned with isolation of endophytic aquatic hyphomycetes fungi from Himalayan regions and to explore their role in ecosystem functioning, plant health, and bioprospection of novel metabolites. Dr. Pant has published a number of papers in journals of national and international repute and has also one book in her credit. In addition, her research is involved in teaching undergrad and postgraduate students and also research mentoring at university level. She was a recipient of a Young Scientist Award by the Indian Mycological Society in 2018, and her long-term research vision is centered on advancing mycological research for biodiversity conservation, sustainable agriculture, and the discovery of novel bioactive compounds from fungi. Thank you so much for joining us, and I hand over to you, Prabha. Thank you.
Thank you so much. Thank you so much, dear Isabel. Now, first of all, I would like to sincerely thanks to the organizing committee for giving me the opportunity to present my work. It's, I can say it's really an honor to be part of this event, and I truly appreciate the efforts and support of making this platform available for everyone. So, without wasting time, I will definitely share my presentation with you all. I think it's visible to everyone.
Yes, perfect.
Okay, so my topic is endophytic aquatic hyphomycetes, diversity and bioactive potential. So first of all, what are aquatic hyphomycetes? Aquatic hyphomycetes or freshwater perennial fungi or ingordian fungi, we can say, these are basically the unique group of perennial fungi which basically belongs to Deuteromycotin. And these fungi mainly discovered by Professor C.D. Ingold in 1942. These fungi, they are basically recognized for their decomposition role, and they mainly present in the leaf litter in freshwater states. These fungi, they are mainly characterized by their characteristic spore types, as you can see in this diagram, that these are basically having different types of shapes like sporosporus, polyposporus, helicosporus, etc. These fungi, they basically participate in the decomposition of leaf litter in stream ecosystem and mediate nutrient flow in stream ecosystem. Although these fungi, they are basically recognized for their role in decomposition, and we can see they are basically the saprophytic fungi, but now these fungi, they are also recovered as an endophyte from the submerged and riparian plants. Due to their endophytic natures, these fungi, they are capable to produce various secondary metabolites, just like phenols, alkaloids, steroids, polyketides, etc., which mainly play an important role in terrestrial as well as in aquatic ecosystems. So, how can we isolate these fungi? So, this is just a pictorial representation. I will elaborate this that first of all, what we do, the plant samples, these were collected from in and around the freshwater bodies of any freshwater ecosystem. And these collected plant samples, they were placed in sterilized bags and brought to the laboratory for the further processing. In laboratory, these collected samples, first they were washed with running tap water for 1 to 2 hours to remove the adhering soil particles and other other extra materials. After that, we do basically the surface sterilization. For the surface sterilization, due to this step, the surface sterilization, that is most important step, because In this process, basically, we treat these samples with ethanol as well as with the help of sodium hypochlorite solution. Due to these surface sterilization, basically the epiphytes and all the attached microbes, they will be easily removed. Now, at last, these samples, they were rinsed with the sterile distilled water and blow dry with the sterile filter paper. Now, these samples, they will be incubated for the isolation of endophytic aquatic hyphomycetes. These samples, they will be incubated at 20 to 25 degrees Celsius for 5 to 10 days in an incubator or at a room temperature. Now, after 5 to 10 days, when the fungal hyphae, basically we incubate these samples by two methods. First one is a direct incubation method and other one is a agar plating method. In direct incubation method, what we do basically, we place these samples in sterile distilled water, and in agar plating method, we basically place these samples in a Petri plate containing the malt extract agar medium or potato dextrose agar medium. Now, after 5 to 10 days, when fungal hyphae emerge out from the tissues, these fungal hyphae, they transferred these hypertics, they transferred in the another malt extract agar plates to perform the, or we can say to obtain the pure culture of aquatic hyphomycetes. Now, after when we got the pure culture, basically we will do for the identification and the maintenance of these pure cultures. So how will we identify these fungi? These fungi, because they are basically they have the characteristic spore types, so their morphological identification is also very easy due to their characteristic spore types. But now we have also did that the ITS sequencing, DNA barcoding, and phylogenetic analysis. So these are some methods by which we can easily identify these endophytic aquatic hyphomycetes. Now come to the diversity of endophytic aquatic hyphomycetes. Altogether, around 70 species of aquatic hyphomycetes till now, they were recovered as an endophyte from the submerged roots and riparian plants. While exploring the endophytic fungi and riparian plants, 4 species were recovered as a new endophytic species. One more thing, these endophytic aquatic hyphomycetes, they are not substrate specific. So basically, when we take different type of plant, host plants. So majority of the endophytic aquatic hyphomycetes species, they were isolated from angiosperms, followed by mosses, pteridophytes, and the gymnosperms. Now, it's just a pictorial representation where we can see that the aquatic hyphomycetes as endophytes and their role in nutrient cycle. This slide basically highlights the dual ecological role of these fungi functioning both as a decomposer in leaf litter, as in aquatic system we can say, and as an endophyte in terrestrial plant ecosystem. In saprophytic phase in aquatic ecosystem, we can say they break down basically the leaf litter and with the help of the extracellular enzymes and release nutrients in the stream ecosystem. While as an endophyte, we can say they enhance the nutrient absorption efficiency, improve plant growth, and also helps in the stress tolerance. So why it adapt the dual life cycle, we can say. Why? So there are lots of various reasons that the aquatic hyphomycetes is adapted the saprophytic phase and endophytic phase. So with the help of the moist soil, these fungi, they may travel towards the roots of the riparian plants and colonize the internal tissues of the plants. And after some time, when the conditions will be favorable for these fungi, they again disperse in the submerged water and colonize the leaf litter in the freshwater ecosystem. And these are some spores of the conidial aquatic hyphomycetes fungi. Now come to the most important thing, that is the bioactive potentiality of these fungi, these endophytic aquatic hyphomycetes fungi. So there are various endophytic aquatic hyphomycetes fungi which are reported for their antimicrobial activity, nutrient solubilization potentiality, plant growth promoting potentiality, as well as antioxidant activity and enzymatic activity. In antibacterial activity, these are basically they inhibit the growth of plant pathogenic fungi. There are various species which have the antibacterial potentiality. and antifungal potentiality. There are some reports in nutrient solubilization potentiality of these fungi like Tetracladium satyserum, Campylosphora purpura, Helicomyces, and Varicosporium melody. There are a number of 6 to 7 species of endophytic aquatic hyphomycetes fungi which are responsible to solubilize the phosphorus and potassium to convert that part in insoluble to soluble form and provide the plant. There are also some reports that these fungi, they are capable to promote the plant growth in vitro conditions. Another one, there are some reports, only 2 or 3 reports, that these fungi, they are capable to produce these some antioxidant compounds, and because they are saprophytic, so they basically produce different type of enzymes and they show the enzymatic activity also. So there is one another list that is the major bioactive compounds from aquatic hyphomycetes and their bioactivity. So the first compound, that is the angulosporose, which has a polyketide in nature that is isolated from the Angulospora longissima, and that it has an antimicrobial activity, and that compound, it was isolated by the Harrigan et al. In 1995. Another one is a tannalic acid A2D that was isolated from Dendrospora tannella. It has also the antimicrobial and cytotoxic activity. Another one is cleveropsin A and B and cleveropsin C. These compounds, they have an antifungal activity and they are basically the cyclic depsipeptides, and they were isolated from the Cleveropsis aquatica. Another one is tricladolides A2D and tricladic acid. They were isolated from the Tricladium species and they show the antifungal activity. Recent analysis on GCMS analysis of 2 endophytic aquatic hyphomycetes species, Tetracladium satyserum and Campylosphora purpura, revealed the presence of various bioactive compounds such as steroids, phenols, flavonoids, etc. And these compounds, they have number of bioactive potentialities, like they have antimicrobial activity, antioxidant activity, and some they have cytotoxic activity also. In the case of Tetracladium satyserum, we have isolated round of more than 30 compounds which have various bioactive potentialities. And another, in the case of Campylosphora purpura, there are 14 different type of compounds we have found in the ethyl acetate extract of Campylosphora purpura, which shows the different type of activities. It represents that these fungi, they are capable to produce different type of bioactive compounds and which show the different type of bioactive potentialities. So basically, what is the trend in research on aquatic hyphomycetes fungi? Research on aquatic hyphomycetes fungi, endophytic aquatic hyphomycetes fungi has expanded now. Once we thought that these fungi, they are basically the decomposer, but now they are increasingly studied for their terrestrial life cycle due to the production of various type of bioactive compounds. Which basically these bioactive compounds, they play an important role in agriculture and pharmaceutical sectors. Previously, these in 1950s, 60s, 70s, and 80s also, it basically their dual life cycle that was recognized, and diversity, taxonomy, and biogeography of these fungi, they have been recognized. After that, these endophytic aquatic hyphomycetes, their role in nutrient cycling, ecosystem functioning, plant fungus interaction, and their role in plant health, that how they increase the growth of the plant, how they solubilize the different type of inorganic compounds and provide that to the plants, and how they protect the plant from different biological and abiological stress. And now basically biotechnological and applied potential of these fungi, it has been explored. So future prospect of these fungi, endophytic aquatic hyphomycetes are, we can say that these fungi, as compared to terrestrial fungi, terrestrial endophytic fungi, these are still underexplored compared to the terrestrial endophytes. Because of their dual ability, we can say that saprophytic and endophytic. In saprophytic, they basically decompose the leaf litter and inhabit the plant living tissue in endophytic nature. We can say that their future prospect basically lies on the eco-friendly plant growth promotion, biocontrol agent against the pathogens, and the synthesis of proven bioactive compounds. We can say in sustainable agriculture and crop production, these fungi, they may be used as a biocontrol agent, stress tolerance, and they also help in the nutrient solubilization. So they basically act as a biofertilizer. In bioremediation, they basically, due to their ecological adaptability in aquatic and terrestrial system, they basically bridge the gap between aquatic and terrestrial ecosystem, and they also help in the bioremediation also. And by the extraction of different type of bioactive compounds, these fungi, we can say that these are the rich source of the novel bioactive compounds, and due to the extraction and isolation of novel bioactive compounds from these fungi, they may be used in the pharmaceutical industry. So, thank you so much.
Thank you so much, Prabha, for making us this short introduction about this other side of this group of aquatic fungi that sometimes we don't really link to terrestrial ecosystems, but they also have this role to play as endophytes and all this potential in terms of their biochemical potential. So let me just quickly check if we have any question. Yes, we have a question for you. So maybe I would just ask you if you could just stop sharing your screen so we can see you better, and also our participants. We have a question from Gustav Redel that asks, have any of these hyphomycetes been isolated from grasses, and has any plant pathogen inhibition been shown in planta?
Yeah, there are, we have basically in our laboratory, we have isolated around 6 to 7 species from the grasses also, herbs, plants also. Yeah, there are some species, basically the species of Angulospora, Tetraclarium, and they have been isolated from the grasses species.
Very interesting. Thank you so much. I remind our participants that if you have other questions, please put them in the Q&A. Because we just have, I think, one question in the chat that I will just read quickly that says, does culturing these fungi on plates present any difficulties? How did you overcome them?
Yeah. There are lots of difficulties to culturing these fungi. Basically, the maintenance and culturing of these fungi is very difficult. For the minimization of the bacterial species, we can say we basically add the little amount of streptomycin and other antibacterial agents also. And there are the random subculturing and random observation due to random observation by random observation and by random subculturing and culturing of these fungi. We can culture these fungi. But during my work, I have basically isolated around of more than 25 species, but I'm able to culture only 5 to 6 species of these fungi because their culturing is very tedious and very difficult.
Mm-hmm. Thank you. I think I will just read one last question, and then for the sake of time, we need to move on. We have a question by Amy Chong that says, just out of curiosity, can endophytes grow independently of their plant hosts to obtain their enzymes?
Sorry, please repeat.
Yes, the question is, can endophytes grow independently of their plant hosts to obtain their enzymes? That's the question.
Yeah, it may be, but there were not that proper reports available, but it may be. I think it may be.
Okay. Thank you so much. I think we need to move to the next speaker. Sorry. So I hand over to Jeff. Thank you.
Thank you.
All right. Thank you so much, Dr. Pant and Isabel. And yes, it's my pleasure to introduce the next speaker, Dr. Ziming Wang. Dr. Ziming Wang is a postdoctoral researcher at the Department of Aquatic Sciences and Assessment of the Swedish University of Agricultural Sciences. He works with the Fun Action Project, leading the leaf litter decomposition experiment, and assessing whether the current strategy and designated protected areas effectively preserve functional services provided by aquatic fungi. Originally from China, Ziming finished his higher education in France. He obtained his PhD at the University of Lorraine in 2023, and he focused on understanding the nutrient requirements of freshwater microbial decomposers, including freshwater fungi and bacteria, for optimal growth and decomposition of organic matter in freshwater systems. Ziming is writing his own research project, hoping to establish a stoichiometric landscape of aquatic fungi. And please go ahead when you're ready, Ziming.
Thank you, Jeff, for the presentation. Hello, everyone. So I'm going to talk about the stoichiometry of aquatic fungi. and why are they important for our aquatic ecosystems. And because we have a such broad audience here today, instead of straight going to the aquatic fungi and the stoichiometry, I want to start with a simple analogy. If the slide wants to change. Thank you. So let's start with coffee. Let's pretend we all drink coffee and we all have our go-to choice for coffee. But for the sake of this example, let's consider only milk and sugar. So all of us will come up with a chart, something like this, with our addition of milk and addition of sugar. So each of points represent each of us and show us how much sugar and milk we want in our coffee. And so each point shows our perfect blend of coffee. It's our best coffee choice. And what do we do when we have our best coffee? We get super productive because our battery is charged. Now, some of you might say that, well, I accept my coffee to be a little less milk or a little bit more sugary. So we can also show that kind of range in this type of figure. So we see we have our perfect range that our battery charged, but we also have a acceptable range where we say, okay, more or less it's doable. But anything outside of this range, we'll say, well, the productivity is not there. We're not going to be able to do any kind of job. So we see that our productivity basically can be measured by milk and sugar in this case. And by studying this balance of milk and sugar, and often it's represented in a ratio, and how this balance can affect our productivity. So by studying this balance, we can understand what makes the best productivity for us and what range is acceptable for us. And each person can more or less be characterized by a different ratio and range. So in ecology, we study the balance of elements and how this balance affects a living system. So this living system can be as small as a single cell or as big as a whole ecosystem. So this study and the balance, so that's the study of this balance and this balance is called ecological stoichiometry. The reason that this works is because biological, ecological functions, processes, and also interactions are involved in different biochemical components. And those components are made of elements, carbon, nitrogen, phosphorus, etc., in different proportions. And aquatic fungi, as well as other living organisms, including us, are also made of elements in different proportions. But nitrogen and phosphorus are the two major elements that most studies have focused on, other than the carbon. So we can see that in this figure, that the average nitrogen and phosphorus content for some of the major biomolecules. For example, protein here, in average doesn't contain any phosphorus, but nucleic acids in average contain have a nitrogen to phosphorus ratio of 15:9. So now if you try to grow some fungi on the culture media with different and nitrogen to phosphorus quantity and ratios, you will see that the growth rate will be different. So here we have 4 different aquatic fungi, or more specifically aquatic hyphomycetes, and we grow them on those 4 different type of nutrient medium. So they can have either more phosphorus or more nitrogen, or in the middle you have 2 different quantities of balanced phosphorus and nitrogen quantity and ratio. So we see that in this case, that the difference in growth rate are mostly decided by which species they are. And the effect of a single medium type depends, the effect of the medium type on growth depends on the species. So each of those species prefer one of the 4 tested medium type with a different nitrogen phosphorus ratio or quantity. But size is not the whole picture. So we've also measured the mycelium density in this study. And we see that the abundance of nitrogen and phosphorus quantity and also a balanced ratio of those two allow the fungi to grow more densely across all the 4 tested species. And if we look at the both figures together, we can notice that the different species have distinct strategy on the given medium type. So it is the case here for Nelu that they grow faster but very thin. Compared to this TRSP species, they grow very slowly, but they are very, very dense. So you can think of this as 2 people drinking coffee and clean their house. Nelu here started cleaning a whole house at the same time, but the TRSP started, for example, only in the kitchen. And then at this point, you might ask, well, how the growth of some aquatic fungi have anything to do with aquatic ecosystem? Well, because those fungi live, as the name suggests, in freshwater streams. And because freshwater streams, especially the smaller ones upstream, are detritus-based ecosystems. Those streams are often covered by riparian vegetations because they are very, the size of the stream bed is very small. So the sunlight can very hardly reach the water to promote any primary production inside the water. So any kind of plant, water plant can hardly grow in this kind of system. So instead, detritus, so the plant eaters, fallen leaves from the trees surrounding them, are the main type of carbon and energy source in this kind of ecosystem. And aquatic hyphomycetes are the main decomposers of leaf litters in freshwater streams. So those fallen leaves are rather rich in carbon compared to the nitrogen and phosphorus content. And our aquatic fungi requires a bit more nitrogen and phosphorus relatively to the carbon content. So those 2 numbers, 2 ratios you see here are very, very generalized, so don't take them literally. So you see there's a big difference in the carbon ratio, which means that the extra nitrogen and phosphorus in this case for the growth of aquatic fungi will have to come from another places, and often they came from as a dissolved form from the water from the stream. And there are a lot of studies that tackles this carbon, nitrogen, phosphorus ratio and how important they are to the aquatic fungi and the decomposition. So if you are interested, you can read some more here. And here is a quick example of how nitrogen and phosphorus ratio can affect decomposition. So this is a study where we measured the cellulose decomposition by 4 different decomposer communities, including aquatic fungi and also bacteria, under 10 distinct nitrogen to phosphorus ratio. And we see that the cellulose decomposition depend clearly on the ratio tested, and each of the community will have a different optimal ratio which maximize their cellulose decomposition efficiency. So efficient decomposition by aquatic fungi is essential for maintaining a healthy freshwater ecosystem. And the beneficial outcome is not only for the environment, but also contribute to the society and the economical growth of our society. And because those fungi are not visible by naked eyes, they are silently doing their job without anyone noticing, but we are here to raise the awareness around among publics. And so people can understand the importance of them and start to care about them. And to finish, here are some take-home messages. So we see that aquatic fungi are made of nitrogen and phosphorus in defined proportion, but also including other elements. They have distinct growth strategy on the nitrogen phosphorus ratio. We see that aquatic fungi are the main decomposer in freshwater, and the imbalanced nitrogen phosphorus ratios can negatively affect the decomposition process. And here are something that I didn't talk about, but I want to mention here is that temperatures, pollutions, all kinds of environment factors can change this ratio and or directly affect the aquatic fungi. And in turn, they will affect the ecosystem service they provide in this case. And with that, I want to thank FunAction and other partners to give me this opportunity to present today. And thank you all for listening. Thank you very much.
Thank you so much, Ziming, for your presentation. This is really great. I remind people that they can put their questions on the Q&A. And I think we have one already. Jeff, maybe you can take over this question?
Yeah, happy to. So let me take a look here. So the first question is from Gustav asking, are aquatic fungi the food source for any other organisms?
Yes, definitely. They are, for example, the preferred food choice. Well, I guess aquatic fungi plus the substrate they are decomposing are the preferred food source for invertebrates, so larvae of insects that are living inside the water, because they bring because as I said, the leaves are very, very rich in carbon, but not so you think that as a dark coffee without milk and sugar, so it's not very drinkable. So yes, they are food source for other organisms. And those insects would eat those aquatic fungi, and in turn, the fish would eat those insects. So it's in food web, they are the basic of how the nutrient would go up back into the trophic level. So yes.
Great, thank you, Ziming. I don't see any other questions in the chat, but Paula has a hand raised, so I'm going to allow you to talk if you did want to speak your question. Please go ahead. Looks like maybe there's not a question there, but there is another one in the chat. So Mark asks, how do nitrogen phosphorus ratios influence the sexual morph of aquatic fungi?
So aquatic hyphomycetes, though, it's a group of it's a ecological group of aquatic fungi, and we observe them mostly in the asexual form. So I have no idea about the sexual, how the nitrogen phosphorus can, well, I'm sure they can in fact affect the sexual morph, but I have no idea in what way they would in fact affect them. Yeah.
Okay, we may have time for one more question. Thank you. So sorry if I'm saying your name wrong, but Pratiksha, you have your hand raised, so I'm going to allow you to talk if you'd like to speak your question to Ziming. Pratiksha, do you have a question?
I'm sorry, it was by mistake happened.
Okay, thank you. Okay, final question then from the chat. Will the community, I'm not sure if that's in or on the assemblage, also change according to the concentration of nitrogen and phosphorus? This question is from Enzo in the questions and answers.
Will the community at the assemblage also change according to concentration of? Yes. So this also something I guess I jumped over is if you have a community of aquatic fungi or with bacteria, then imagine each of them would have a preferred nitrogen and phosphorus ratio, and the community would adjust the presence, so the abundance of each of the individual species to make up for the whole community optimal. So yes, based on the ratio of each individual species, they will be favored or disfavored when they are present in the given ratio.
All right, thank you very much, Ziming. And
Thank
I
you.
believe Isabel will be introducing our next speaker.
Thank you so much. Yes, and our next speaker, oh, my English today. Our next speaker is Professor Patricia Fiuza. Professor Fiuza, she works at the Institute of Biology of the Federal University of Bahia in Brazil. She obtained a bachelor's degree in biological sciences from the State University of Feira de Santana, followed by a master's degree and a PhD degree in botanical sciences from the same institution. During her doctoral studies, she undertook a sandwich PhD program at the Coastal Carolina University in the United States, with research focused on the taxonomy and ecology of aquatic fungi. Moreover, Dr. Fiuza completed her postdoctoral training in cryptogam taxonomy, botany at, excuse me, I lost myself here. And she currently serves as a curator of the asexual fungi, freshwater fungi section of the International Initiative Outline of Fungi. She also is a member of the IUCN Species Survival Commission Aquatic Fungi Specialist Group and of the Brazilian Fungal Specialist Group, where she coordinates the Caatinga Working Group. In addition, she coordinates the Laboratory of Microfungal Biology. Thank you so much for accepting our invitation. The floor is yours.
First of all, I would like to thank you for the invitation. I am so happy to be here today. And speaking about the aquatic fungi in South America. So my lecture is about the hidden biodiversity in South America waters, richness and distribution of aquatic fungi. South America covers an area of approximately 18 million kilometers and comprising 30 countries. The continent harbors remarkable biodiversity and endemisms with some hotspots like Atlantic forest, Cerrado, the Valdivian temperate rainforest, the tropical Andes, and the Tumbes-Chocó Magdalena. It is a river continent with many food plants, lakes, lagoons, and some rivers. I start here the Amazon River, that is an Amazon basin, the largest drainage river of the world. So the South America has high potential to reserve these aquatic fungi. Despite of this, South America continent is suffering with anthropogenic impacts, including the deforestation, the wildfire, and the pollution of waters that can be lethal to this fungi. Aquatic fungi complete at least one part of the cycle in the water and distribute spores above or between water. And they are so important to the cycle of the nutrients in this ecosystem. To date, we have 3,870,000 species described in this ecosystem, and this fungi has diversity phylogenetic, including 9 phyla, between them Pitridiomycota, Glomeromycota, Basidiomycota, and Ascomycota, that is the richest in between this. The vast majority of these species are inland and unexplored. Speak about South America, the first work developed was by Nilsson in 1962, that described 2 species, Angulospora aquatica, that is an endemic species from the South America, and Pyramidospora casuarina, that is known in a lot of tropical waters from the Venezuela. In Chile, Burgos and Riffart in 1982 were the pioneers of the study of aquatic fungi, collaborated later with Dr. Scherer And I need to highlight too the work of Matsushima that conducted extensive collection in the Amazon areas of Peru and Ecuador in 1980s, describing more than 60 new species. And in Brazil, the studies began in the late of 1980s with the work of Dr. Iracema Schulain Crucius, that was the first to reveal the diversity of aquatic hyphomycetes in South America. According to our database, South America shows 700 species of freshwater freshwater fungi, including 191 ascomycetes and 9 basidiomycetes, of which 189 are Ingoldian fungi. How we can see in this map, we have the actual distribution of the studies with aquatic fungi. In South America, just Guyana, Suriname, Bolivia, and Paraguay doesn't have studies focusing in aquatic environments, but the Brazil has the highest number of studies and the highest number of species. Now we comprise 486 species. Currently, my team with Dr. Flávia Barbosa from the Federal University of Mato Grosso are working in Amazon areas. The most of our species are from Sordaromycetes, followed by Dotidiomycetes and Leotiomycetes. In this year, we released the first chapter of the work of my PhD student, Heloísa Farias, that revised the diversity of freshwater Dotidiomycetes in the Amazon. This work was done based on the literature and the data of these fungi in the herbarium. Mainly two herbarium in Brazil, that in Belém and in Feira de Santana, in Bahia. So in this work, we got 153 freshwater Dotidiomycetes and Pleosporales and Tuberophiales were the most represented. We also analyzed the distribution of families of Dotidiomycetes in the Amazon biogeographic areas, and we conclude that Belém, Xingu, and Inambari were the areas with the most species of Dotidiomycetes, highlighting here Tuberophiaceae. Belém and Xingu are located in Brazil, and Inambari located in Peru. This paper highlighted too the beauty of the fungi we have in the Amazon basin. In Atlantic forest of Rio Grande do Norte, we could identify and describe 77 species of these aquatic fungi and highlight some fungi that are here for with just one record, like a Flagellosperma minuta, and we recorded the first time the Tricelophorus conagensis for Brazil. Recently, still in the Atlantic forest, in streams of the Espírito Santo, we analyzed the taxonomic composition of Ingoldian hyphomycetes in streams located in these forest fragments and in the areas altered by lands. So we found 12 species of Ingoldian hyphomycetes, highlighted here again a new rare species that is Trihamolispora duobigbrachiata, and another new records to Brazil and Espírito Santo. In the most of South America works, we can find that many species are classified yet in the certain seeds. And the most species are described just with the morphological characters. So we have a few works that include both morphological and the molecular characters. So I destack here the Roselimices brasiliensis that was described in honor to Roseli Ana Piccolo Grande, that was the first conidial fungi research from Brazil, and co-authored the paper with Iracema Schwellen-Crucius. So with the molecular work, we could place these species in Sortariomycetes, and we highlight too that it is a Brazilian endemic species that was assessed in third Brazilian IUCN workshop for the assessment of endemic species of fungi. So it was the first freshwater fungi from Brazil to be included later in the red list of IUCN. With concluding remarks, these studies demonstrate the remarkable richness of freshwater fungi in South American ecosystems and emphasize that much of this treasure remains undiscovered, highlight that we have to conserve these ecosystems. As a future outlook, we expect to expand with more collection, not more in some biomes that you are not knowledge about the aquatic fungi, but some biomes that we have few studies too. We planning to train more human resources and work more with the molecular approach. Now we have participating of the National Institute of Science and Technology for fungi in Brazil, and we invest more about the placement of this fungi and the studies of metagenomic. So we expect to analyze more species for red list. Thank you very much for your attention.
Thank you so much, Patricia, for this presentation and overview on the biodiversity of aquatic fungi from South America. While we wait for maybe some of the participants to direct one of the questions to you, I think I will ask already a question. So you have been finding so many species in South America, but I can imagine that many more are there still to find, because South America is so big and there is so many places that have not been explored yet. From your experience, are there many samples where you find either through morphology or some of your isolate species that seem to be new species to science?
Yes. In all the works we have done, we have rare species, new species, and now we are investing more in isolate this fungi, try to do at least the DNA barcoding to position this fungi and bring more works with this integrative taxonomy. That is important. So in all this biome, we can find more and more new species all time. So in Amazon is impressionant about this, so we can see after some spoilers.
Maybe as a continuation from my question, we have a question from Margot Jans that says, maybe I did not hear it if you mentioned, but what other molecular approaches do you have in mind? For example, metabarcoding or environmental DNA may be a solution to find more species?
We are begin the work with metabiology and the eDNA in this year.
I guess, do you have an idea of the genetic databases, if they are well covered for the species of South America, or if we need to invest a bit on producing the DNA barcodes for the South American most common species?
Oh, so few studies, so few, like 2 or 3 studies in South America. So we have the investments of the work with metabiology first in 2012. And how the molecular work is so expensive for us, it is costs a little time. So.
I think I take just one last question from Gustav that says, given the number of ecosystems in South America, how many have you sampled? And have you found species that are very geographically limited, restricted in just one area?
So we have some species restricted, like 12 species cataloged in this. And about the samples, we have a high distribution of these points, but it's like the studies are scarce in some points. We can monitor it all the time, these points. So because of this, we need to do more collections.
Thank you so much. I hand over to Jeff.
All right, thank you very much, Dr. Fiuza and Isabel. It's my pleasure to introduce our last speaker, Dr. Jeffrey Zahn. Dr. Zahn is PI of the Tidal Lab at William and Mary. He's a community ecologist and data scientist who studies the global distribution and assembly of microbiomes using large-scale sequencing and machine learning. He's contributed to and led multi-collaborator programs like the Global Soil Microbiome Consortium, a long-term ecological research Spark project, and the National Microbiome Data Consortium, generating data sets that map fungal taxa and traits across ecosystems and continents. His work combines metagenomics, neural net modeling for predictive ecological and evolutionary integration, microcosm experimentation, and trait-based approaches to examine how fungal communities assemble and shift across environmental gradients. He's also developed bioinformatic methods to reduce misclassification in large metagenomic data sets at a global scale. Welcome, Dr. Zahn.
Hey, thank you for having me. Let me get my screen brought up here. All right. So I'm going to talk briefly about marine mycology, the fungi that live in the ocean and associated habitats. There we go. We've known about these things since the mid-1800s. The first marine fungus was described, but we haven't done much with it since then, honestly, until very recently. And I like to think of this field as kind of being old-time explorers charting the globe and drawing maps. And what we've got here in the top left is a map of the east coast of the United States, what is now the United States, America, in early 1500s, and you can see it's pretty weak. There's a lot of uncertainty, it's low resolution, and maps often did this back in the day before the whole globe had been mapped in any detail. They just kind of trailed off at the edges. Those are the margins of the maps, and one famously said, "Hic sunt dracones," here be dragons. And they weren't really saying that there's anything to be afraid of necessarily. It's just this kind of placeholder of uncertainty. We don't know what's beyond here. And that's kind of how we've been treating marine fungi. Like biological curiosities, kind of best avoided. If you want to get any real work done, stay away from them. There's not enough known. But you can see that we can find marine fungi, and they've been around for a long time, but we still are kind of arguing about how to define them. Because as you can imagine, fungal spores are blowing in through the atmosphere, being deposited by rain, and they land there. And if we find those fungi, they may not be marine fungi. It might just be a tourist, where it's just a spore sitting there. And so we're moving towards metabolic definitions, those things that are metabolically active and can complete their life cycle or a big part of their life cycle in the ocean. And this includes a lot of different habitats. The ocean's a big place, from the deep sea vents all the way onto the mangrove coastal environments. And yes, we do find fungi living on the carapaces of crabs down in the deep ocean vents, well above boiling temperature and high pressure. We find them in the open ocean. We find chytrid fungi parasitizing algal outbreaks. We find them inside the leaves of seagrasses and in salt marshes and in mangroves. They're found all over, but they're kind of hard to study. They tend to be microscopic. Like all aquatic fungi, these aren't really making nice, pretty mushrooms that people are used to thinking about when you think of mycology. They can be really hard to culture in the lab too. As you've heard, some of these are tough to grow. These, like saltwater, some of them require high pressure to grow. So culturing them can be a real challenge. And historically, oceanographers hasn't been studying these, and that's who's been doing a lot of the work on oceans. And so eDNA won't save us in this case. And that's because there's not good enough representation in our genomic databases. And so it's pretty common finding for me to get a sample from some marine habitat, do some deep metagenomic sequencing and assemble contiguous DNA sequence and try to say, okay, what is this? And it just comes back and says fungus, ascomycete, unknown everything else, right? So this is almost certainly a new species. It's nowhere in our database. We can tell that it's fungal, but not much more than that. Right now, only 7 of the 2,500 microcosm fungal genomes are from marine habitats. And so our database currently sits at, I think, around 40 marine fungal genomes that are in any shape at all, let alone complete genomes. Compare that to bacteria, we've got more than 2 million fully sequenced and annotated genomes available in our database. So 40 versus 2 million, it makes this a bit challenging, where eDNA is commonly detecting fungi, but we can't say who it is, and we don't know much about it. But we've had some recent good news, and a paper from last year by Eva Breyer and others really tried this triangulating approach at trying to get at fungal biomass in the marine ecosystem. And they were looking at open ocean, these pelagic systems, and they used 4 different methods calculating fungal biomass, and were able to basically get the same result, which reduced our uncertainty quite a bit. And this is brand new. Before this, we had kind of wild guesses with high error bars about the biomass of fungi in the ocean, but we've narrowed it down at a lower limit of 0.32 gigatons. And so that's 9 times the amount of archaeal and about a fourth of bacteria. And so these are major players, and the map is starting to get filled in a little bit here. We're getting a little bit more definition, definitely more clarity, less uncertainty in our estimates of how many fungi there are in the ocean. And this is just the pelagic habitat. This is important because now we know that they really are major players in the marine ecosystem. For one example here, to a comparison, 0.32 gigatons of carbon is 22 million blue whales. So this is a lot of biomass, and fungi from all ecosystems are known for having really important roles to play in nutrient cycling. in particular carbon. And so we can look at the oceanic food web, and we've got sunlight coming in and phytoplankton fixing carbon from the atmosphere and dissolved CO2 kind of being the base of this food chain. And we've got parasitic fungi that are absolutely kind of following these bloom and bust cycles of phytoplankton. But we've also got saprotrophic fungi, which are breaking down nutrients, kind of passing them in. We have marine snow. You can often find fungi sitting on marine snow. And so there's this kind of fungal shunt that's moving carbon through the ecosystem in ways that we kind of have been ignoring, especially in our large global modeling efforts. And so there's ongoing efforts to kind of utilize this newly congealed knowledge where we're certain that these are big players, but where are they and what are they doing and who's actually there? These are open questions. And so efforts like improved culturing approaches, the culturomics is one way forward, where these are hard to grow. And so we've developed new ways of trying to culture them in situ. And so methods like microfluidics and the iChip, where we're taking some marine sample and diluting it, hopefully to where there's only one cell per microliter, and then plating that out on these 96 well plates. You put a 0.2 micron filter over that and then stick it back into the ocean. Because we might not be able to grow these in the lab, but there's something there in the environment that they're depending on. And so we can get them to grow up in the environment without contamination that way. When we do these sorts of things and we get tricky fungi from marine habitats actually growing in the lab, we start experimenting, we're discovering that these are absolute gold mines for all kinds of secondary metabolites. So all kinds of new promising antibiotic compounds. We've found proteins that can degrade microplastics, and the Amen lab has even been experimentally evolving these to eat plastic faster, right? So there's great biotechnology just at our fingertips hidden in this diversity. But we're just getting started. We're at this inflection point in marine mycology where things are kind of coming together, and it's a really exciting time to be in this field. We've got more precision in our estimates of how many fungi there are. We have new ways of observing them in situ and seeing who's there and what they're interacting with. And we've got converging incentives where more and more people are paying attention to climate change and microbiomes and the ocean. And so this is a really exciting field where pretty much every time we go in and culture something, it's a new species. That makes it really annoying for somebody like me who's interested in doing metagenomes, eDNA work to kind of see diversity because it just comes back unknown. But that's also really exciting because we can go out and get a liter of seawater and find 10 new species in it. Okay. But the map is getting sharper, and as it does, our questions are becoming, I think, bigger and more interesting. And so thank you for letting me hopefully get you excited about marine fungi. And if you're interested in following any of our work, my website is down here at the bottom. So thank you.
Great. Thank you so much, Dr. Zong. Let me see here. There is a question in the Q&A. It looks to be quite long. I'm just going to start reading it out here. So it's quite common to face problems with specificity of card fish probes for fungal groups, as the organisms are quite diverse and sometimes even CWF stains other eukaryotes. In addition, we have a gap on how some fungi, besides well-studied chytrids and hyphomycetes, look like in aquatic systems. Do you have a recommendation for the best approach nowadays to evaluate fungal biomass and link it to activity in aquatic systems?
I'm probably the wrong person to ask about that. I'm a genomic data scientist that just got really excited by this new finding. Yes, there are limitations in some of these methods that you just pointed out. And so really, I think of this as a low-end estimate of the amount of fungal biodiversity or fungal biomass. In particular, a lot of chytrid fungi would have been missed by these survey methods, and they didn't really intentionally follow algal blooms, right? And so if you look in different parts of the ocean, I think we're going to get different estimates. This is just kind of the low end, but it's nice to have that precision on it, where these four methods all pointed to the same general number.
Great, thank you. Did you want to chime in on that one, Isabel? Or
Yes. So we have another question from Gustav that says, have there been any marine fungi that have been shown to be endophytes of coastal plants? And are there any marine fungi that are culturable in simple media such as extra salty PDA?
Yeah, this is my go-to is malt extract agar made out of old seawater. And yes, all kinds of endophytes in seagrass meadows and in mangroves. And those are the two that I've personally studied, but absolutely all kinds of salt marsh plants have fungal endophytes in there. And just like terrestrial endophytes, they tend to be the easier things to grow in pure culture. And so that's actually really nice because we can smash up some seagrass leaves and put them on a plate of PDA made out of salt water and they will grow, which is great. A major area of my research is trying to engineer better microbiomes for seagrass restoration. And so we're doing that, getting a culture library of those fungi, and it's turning out to be fairly easy to grow those. Yeah.
Thank you.
great.
I see the hand of Ziming up. Ziming, you want to ask a question live?
Yes, I do have a question. When thinking about marine environment, the water pressure can also be an important factor, right? And if you try to grow them on surface with atmospheric pressure, it makes sense. Some of them might not like this low pressure environment.
Oh, absolutely. And some of them are only culturable in pressure chambers for that reason, making it very
cool.
challenging. Yeah.
Thank you.
Okay, I think we should probably wrap up. Is that right, Isabel?
If there is no more questions, let me just quickly check here. Maybe we have time for one more question.
Okay, there's one in the chat. Let's see here. Have you come across any hints of symbiotic relationships between endophytic fungi with their hosts or other microbes within the holobiont?
There seems to be a couple hints of good synergy between seagrass bacteria and fungi, where the effect on the plant depends on both partners being there. I have not started teasing that apart and seeing if how they perform individually, but it seems like the plants really need both for some of those effects. That's a whole other can of worms, the fungal bacterial interactions. Well, thank you very much.
That makes sense. There's a lot to go over. Thank you very much, Dr. Zahn. I'm just going to share the final slide here so we can acknowledge everyone who's helped plan this webinar today, facilitate this webinar, and of course all the speakers for sharing your knowledge in this webinar. So thank you for attending. I'm sure we'll be sending out a follow-up with at least the link to the recording. And I'm not sure if there's anything else you'd also like to share at this time, Isabel, as a closing.
No, I think just thanking again the speakers today because they really showed up how aquatic fungi are so diverse and so important to protect, and to wish everyone happy International Day for Biological Diversity 2026, and hopefully see many of you next year. Thank you.
Thank you.